Welcome to the Future Lab, the podcast exploring the frontier of autonomous science. I'm your host, Matthew. Today, we'll delve into a groundbreaking paper detailing eVOLVER, a DIY platform for high-throughput automated cell cultures. This research tackles the challenge of balancing precise control over experimental conditions with the scalability needed for large-scale studies in fields like molecular evolution. It's a really exciting area, isn't it? The ability to automate complex biological experiments opens up a whole new world of possibilities. We'll explore its innovative design, examine the technical details, and discuss its real-world applications. And, fun fact, the script for this episode was partially AI-generated, highlighting the very technology we discuss. Isn't that a neat meta-commentary on the power of AI? So, what is the core problem that eVOLVER aims to solve? Before the advent of this platform, what were the limitations researchers faced when trying to perform these kinds of large-scale experiments? Essentially, traditional methods struggled to simultaneously achieve both precise control of experimental parameters and the high throughput necessary for large-scale studies. You needed either highly controlled, low-throughput systems like bioreactors, or high-throughput, but less controlled, batch cultures. eVOLVER cleverly sidesteps this trade-off, offering a customizable and scalable solution. This opens the door to a range of new experiments that previously would have been too time-consuming or impractical to undertake. We'll be diving into the specifics of its design shortly, but first, let's get a high-level overview of the key findings of this research paper. The main takeaway is eVOLVER's ability to perform complex, high-throughput experiments. Think evolving yeast populations under multiple selection pressures or screening entire yeast knockout libraries to discover genes involved in things like stress response or adaptation. It's a game-changer for fields like molecular evolution and systems biology. Now, let's move on to the specifics of how they designed and built this incredible piece of kit. So, the paper by Wang et al. Introduces eVOLVER, a truly ingenious system. Let's break down its design. It's modular, right? What does that actually mean in practice? The modularity is key. It means that eVOLVER isn't a single, monolithic device, but rather a collection of independent, interchangeable components. This allows for incredible flexibility and customization. I see. So, you can essentially tailor the system to whatever experiment you're doing, rather than being constrained by a fixed setup. This is a big advantage over many previous automated systems. Exactly. Let's talk about these three main modules: the smart sleeves, the fluidic module, and the control system. Let's start with the smart sleeves. What's the clever design aspect there? Each smart sleeve is essentially a mini-lab for a single culture. It houses the culture vial, along with integrated sensors and actuators for controlling things like temperature, stirring, and optical density. The beauty of this is it gives individual control over each culture. And the fluidic module? How does that work alongside the smart sleeves? The fluidic module handles the liquid handling, adding fresh media, removing waste, and performing dilutions. They use two different approaches: a simpler peristaltic pumping system for basic tasks, and more sophisticated millifluidic multiplexing for more complex fluidic manipulations. Millifluidic multiplexing sounds advanced. What exactly does it entail? That's where things get really interesting. It's inspired by large-scale integration in electronics. They use miniature channels and valves to control the flow of liquids in a highly multiplexed way. This allows for much more complex fluidic operations, including things like dynamic media mixing, automated vial-to-vial transfers to prevent biofilm formation, and even automated yeast mating. That's incredible. So it's not just about controlling individual cultures, but also about controlling the interactions between cultures. This opens doors for whole new classes of experiments. Precisely. And tying it all together is the control system based around Arduino microcontrollers and a Raspberry Pi. This handles the data acquisition, processing, and feedback control, allowing for real-time adjustments to the culture conditions based on the sensors in the smart sleeves. Everything is orchestrated via Python scripts, which means the experimental parameters are entirely user-defined. This programmable nature of the system is a real strength. So, the system's modularity, the sophisticated fluidic controls, and the powerful programmable control system all come together to address the original challenge of balancing high throughput and precise control. Now that we've explored the nuts and bolts of eVOLVER, let's delve into the impressive applications and implications of this technology in the wider field of autonomous science. So, we've explored the ingenious design of eVOLVER, with its smart sleeves, flexible fluidic control, and sophisticated control system. Now, let's consider the real-world impact. What are some of the key applications of this technology? The applications are incredibly broad, spanning various fields. The ability to conduct high-throughput experiments with precise control is transformative. Think about accelerating drug discovery, materials science, or even synthetic biology. In the paper, they highlight several impressive applications. For example, they use eVOLVER to evolve yeast populations under different conditions. This allows them to study how organisms adapt to changes in their environment. That's powerful, absolutely. And another key application is the ability to screen large libraries of genetic variants, such as yeast knockout libraries. This is essential for functional genomics studies, where you're trying to understand the role of individual genes. Exactly. This allows researchers to systematically investigate the impact of multiple factors on a large scale, revealing insights into complex biological processes. Previously, this level of experimentation would have been incredibly labor-intensive and time-consuming. Beyond these examples, imagine the possibilities in other areas. High-throughput screening of novel molecules, optimizing metabolic pathways in microbes for industrial applications, or studying the dynamics of microbial communities. All of these stand to benefit enormously from this improved technology. One aspect that particularly struck me was their use of millifluidic multiplexing for tasks like automated vial-to-vial transfers. This allowed them to circumvent a major problem: biofilm formation. That's a significant advancement. Biofilm formation is a persistent problem in many long-term cell culture experiments, and this automated passaging approach represents a smart solution. And it's not just about solving individual problems, it's about the potential for entirely novel types of experiments. The ability to program complex interactions between different cultures, for example, opens up a huge range of possibilities that weren't feasible before. True. eVOLVER essentially enables a new level of experimental complexity and control. The researchers even demonstrated automated yeast mating. This shows the versatility and programmability of the system. The fact that this is a DIY system, too, is quite revolutionary. More researchers can access and adapt this kind of technology. It's not just about the technical innovation, but also the accessibility of the technology to more scientists. Absolutely. That democratization of access is a crucial aspect. Now that we've seen the impressive capabilities and applications, let's wrap up with a concise summary of the key takeaways and a glimpse into the future of this exciting technology. So, eVOLVER's impact extends far beyond its innovative design. Let's explore the broader implications. How does this technology accelerate research in molecular evolution? For instance, the ability to conduct high-throughput experiments with fine-grained control over environmental conditions is transformative for molecular evolution studies. It allows researchers to explore complex fitness landscapes with far greater detail than was previously possible. And what about applications beyond molecular evolution? What are some of the other significant ways that eVOLVER advances scientific research? eVOLVER's high-throughput capabilities also significantly benefit functional genomics research. Imagine screening a whole yeast knockout library under different conditions, like varying temperature, to determine how gene knockouts affect temperature sensitivity. This level of investigation was just not feasible before. That's an amazing application. It really highlights how eVOLVER enables a whole new scale of experimentation. Could you give us a specific example from the paper to illustrate this? The paper demonstrates this perfectly by varying the temperature and frequency of temperature changes. They were able to identify gene sets associated with heat shock responses and cell cycle regulation. This level of detail in understanding cellular responses to stress is groundbreaking. It's fascinating how they can link these findings back to the underlying biology. This really points towards the power of being able to manipulate multiple experimental parameters simultaneously with this device. Precisely. And it's not just about temperature. The paper also showcases eVOLVER's versatility in tasks like dynamic media mixing, preventing biofilm formation through automated transfers, and even automated yeast mating. All of these highlight the remarkable flexibility of its modular platform. This really underscores the versatility of the system. It moves beyond simply speeding up experiments to allowing for entirely novel experimental designs. What are the broader implications of eVOLVER for the field of autonomous science? eVOLVER represents a significant step forward in autonomous science. It provides unprecedented scalability and control in biological experimentation, enabling researchers to tackle more ambitious projects and ask more complex questions. It's opening the door to more sophisticated experimentation. It's remarkable how this technology is not only enhancing existing research but also creating opportunities for entirely new avenues of scientific exploration. It's definitely a significant contribution to the growing field of autonomous science. Indeed. And that brings us to our conclusion. Let's summarize the key takeaways and reflect on the future direction of this exciting area of research. Let's bring it all together. Wang et al.'s work on eVOLVER represents a significant achievement in the field of automated cell culture. What are the key takeaways from this research? In essence, eVOLVER offers a versatile and scalable platform for high-throughput experiments. Its modular design, combined with sophisticated fluidic control and real-time feedback, allows for unprecedented precision and throughput in studies of microbial evolution and functional genomics. It's remarkable how this system addresses the long-standing challenge of balancing control and scalability in biological experiments. The implications are huge, aren't they? Absolutely. eVOLVER accelerates experimental biology, opening doors to deeper insights into evolutionary processes and cellular responses to environmental stresses. It's a real game changer. And what about the future directions for this technology? What could we expect to see from eVOLVER and similar systems in the coming years? The authors themselves mention the potential for integrating eVOLVER with other automation technologies, such as robotic systems. This would further streamline workflows and enhance the system's capabilities, pushing the boundaries of what's possible in automated experimentation even further. That's an exciting prospect. Imagine fully autonomous labs, capable of running complex experiments with minimal human intervention. eVOLVER is a significant step towards realizing that vision. Precisely. This technology is not just about incremental improvements, it's about fundamentally changing the way we conduct biological research. It's pushing the boundaries of what's feasible in terms of experimental scale and control. And let's not forget that eVOLVER is also a testament to the power of open-source design and collaboration. Making this kind of technology accessible to a wider community of researchers is crucial for accelerating scientific progress. Absolutely. That democratization of access will undoubtedly fuel innovation and collaboration across the field. It's a truly inspiring achievement. So, to summarize, eVOLVER represents a major leap forward in automated high-throughput cell culture, enabling more ambitious research and deeper insights into complex biological systems. It's a testament to the power of modular design, intelligent automation, and collaborative innovation. Thanks for tuning into the Future Lab. This episode's script was AI-assisted, a fitting example of the very technologies we explore. Until next time, stay curious.